Right, hello everyone, and thank you for inviting me to speak today. It's been amazing to hear all the stories already, and good to hear a bit about the clinical side. So my presentation will overlap a little bit with Ram's presentation, but there will be some other exciting bits for all of you to enjoy as well. Right. Aha. So, oh no. It worked earlier, so yeah. So I'm a PhD student from the University of Southampton, and we work very closely with Bournemouth hospitals. So we work with Helen and Renata, and we have a very big CLL research group in which there are 7 PIs that lead different research branches within CLL. So my supervisor is this dashing man. His name is Professor Jonathan Strefford. And we're a group that research on CLL genomics. So specifically with me, I'm looking at the epigenomics of CLL, but we'll get into that in a minute. So we've already gone through this, but leukemia is a word coined in German and comes from Greek words, Leukus and Heimer, so good for trivia and pop quiz. It's defined as a cancer of the blood. And like with all cancers, it's the loss of normal regulation so that you get this uncontrolled proliferation of cells, or you get the event of abnormal cells growing. But before we go any further, let's talk about what the cell is. So the cells, they're the basic building blocks of all living things. So every one of us has over 37 trillion cells in our bodies. So you're sat on a lot of cells right now. So most of our cells in our body hosts hereditary material in the nucleus, as I've highlighted here in this little square box. So what it looks like in the nucleus. So what we have is our DNA. So we've got, does this pointer work? Oh, no, it doesn't. Sorry. Does it work? Yes, okay. So what we have here is a DNA sequence here that coils to form this DNA helix. This DNA helix winds around different proteins that forms our chromatin structure. And this chromatin structure is then wrapped into another tighter structure, I suppose, that forms the chromosomes. So in our nucleus, we have 23 pairs of these chromosomes, and they form, well, that's the genetic instructions on how we are made. But in reality, they don't look as nice as what I showed. It looks like this, it's a bit plain. So we have 46 all packed in our nucleus, and they kind of make up our genetic makeup. So, yes, so what's important with every cell is that it goes through the circle of life, because, you know, it has to start somewhere, has to end somewhere. So it's really important that this cell cycle is regulated properly to ensure that cells grow and, you know, that none of these abnormal cells are given a chance to propagate. So I thought I'll quickly go through the cell cycle very briefly. I'm hoping this animation works. It worked at home. So what we have is a cell. And in phase 1 of the cell cycle, what we're seeing is a duplication of events, duplication of components, I suppose, in the cytoplasm, so it grows. Ooh. And then after this growth, what we have is a checkpoint. And at this checkpoint, there will be proteins that will make sure that this growth has occurred correctly and that there's nothing abnormal about the cell. So then we move on. This is working, this is good. So in phase 2, what happens is we duplicate our DNA. So here DNA is being replicated so that there's enough to make 2 cells at the end of this process. Again, what we see is this expansion in the nucleus, and then there'll be a second checkpoint. And here proteins again make sure that this process has worked properly, and then we can move on to phase 3. So phase 3, we're looking at more cell growth, and then eventually we should have the event of 2 identical cells. So that's the cell cycle, and it will come to play in a minute in my talk later. So, like everything in life, balance is key. You can't really have the components out of balance. So here we've got cell production and cell death. So in a normal healthy body, what you have is cell production and cell death occurring at a similar rate. And then this is what you see in normal cells, essentially. But then in leukemia cells, and not just leukemia cells, cancer cells as well, what you have is cell production going at a faster rate than cell death, and then you tip the balance towards cell production. And then this this gives rise to cancerous cells, or in our case, leukemia cells. The other event is that you have normal cell production, but still less cell depth. So in the end, the scale still has to tip towards cell production, and so you still get this propagation of cancerous cells. So we've already touched on what chronic lymphocytic leukemia is, but just in case you forgot. So we know it's the most common form of leukemia in adults in the Western world. It arises from B lymphocytes, which is a type of white blood cell. And B lymphocytes are an important part of the immune system, and they basically are heroes against known enemies in the body. So here he runs away. And one of the key things about the B cell is that it has this B cell receptor, or the BCR, which allows the B cell to communicate with other cells, but also to present foreign molecules on its outer surface, I suppose, to other cells, so that certain things can take place. So here's a B lymphocyte. It's looking very dapper. What we have here is the B cell receptor that I was just talking about. And here it's bound to a molecule of interest. So that could be a foreign antibody, a foreign antigen, sorry. And then because of this binding, certain things will happen downstream, and then certain events will take place in the nucleus. So Ram's already gone through this, but what I thought I'd quickly show is what happens in an immature B cell and how it goes through the B cell development, and then you get this maturation of the B cell at the other end of this germinal center. So what tends to happen is you get these naive, innocent, childlike, you could say, B cells that go through, and then they get exposed to these foreign molecules. Upon binding to their B cell receptor, you get a maturation process in which we were talking about the heavy gene rearrangements. So this B cell receptor then becomes more specialized to the antigen that it's just bound to. And so then what happens is it becomes a B cell that is now specific to a particular antigen or molecule that it's bound to. So that's what this diagram is showing. So what you get then is a plasma cell, which will do all the fighting, and then the memory B cell that runs around the whole body and makes sure that if it comes across that molecule again, it will quickly do something to cause an immune response. Right, so the Ig genes, which is something that we've now already talked about, but they're genes that code for the immunoglobulin structures. They're a group of genes that help the BCR to adapt to a particular target or the molecule of interest. And they make the I've already said this. Oh dear. But they make the BCR more specific to a particular stimulus by going through several small but programmed mutations. So I thought I'd show this with a diagram with some fancy animations, should they work. So this process is a bit of a cut and paste job. So what you'll have is, because of a certain molecule, certain gene segments will be pulled together, we'll remove the sections in the middle, and then these will be cut and paste and joined together. So here again, we're doing the same thing again, and then again. And so this will form our heavy chain region. And then we'll do the same thing with the light genes, the light chain genes. So same process taking place. And then eventually what we should get is a lovely B cell receptor. So this B cell receptor is the one that I was just talking about, that is on the surface of the B cells, and they communicate with the other cells. And this process takes place in every single cell once it's exposed to some foreign molecule. So this is not abnormal, this type of mutation. But what we do know is that they're very important for the formation of BCR, of the the formation of the B-cell receptor. But CLL cells can arise from before this process takes place or after this process takes place. So if we look back at this diagram I showed you, you can get CLL occurring at this point, before this process has taken place, or it can take place after this process has taken place. And then what we have are two, oh dear, where's it gone? Okay. We have then two different types of CLL, but why are those outcomes so different? And the thing is, we just talked about that process of the B-cell receptor maturing and becoming more specific to a target. And that's mainly what it is. And from these two CLL types, you've heard of now UCLL and MCLL, or mutated CLL and unmutated CLL. And basically what we're saying is we can now identify the origin of that CLL cell due to its mutational status of the IGHV gene. So, yeah, so different studies have come a long way and helped us identify the differences now in the mutational status and its impact on CLL prognosis. So we've identified that mutated CLL, or MCLL, has typically a better prognosis. It's less similar to the IGHV genes before the cut and paste process took place, and it's likely to have derived from memory B cells. And then we've also got IGHV unmutated cases, which UCLL, which are more similar to the IGHV genes before cut and paste, and they're likely to have stemmed from those naive B cells before exposure to antigen binding, as you can see from here. So there's our similarity. So typically, we say that UCLL cases are more likely to follow an aggressive disease course, and MCLL cases are more likely to follow an indolent disease course. However, nothing is this simple in biology. This is how we look all the time in the lab, like, So, on top of that, there are other chromosomes that I was talking about. We have 46 in our nucleus. So we have different problems coming up. So here we have our normal chromosomes. We've got one pair of long chromosomes and one pair of short chromosomes. And then what you can see here is that there will be different mechanisms in which these chromosome abnormalities will cause issues downstream with with the cancerous cells. So we've got translocation events. I'm not going to go through these in detail, but just to let you know that these are what's happening. So we've got inversion events, we have duplication events, we have insertion events, but I think the main thing in CLL is deletion events. So what you have here, in this case, is that this segment here, which should be here, has now been deleted. And that can cause different versions of CLL that we see. So, if you remember to my sexy cell cycle from before, what I highlighted before was those checkpoint proteins that help make sure that the cell hasn't done something that it shouldn't have done. So, these proteins are essential to making sure that the cell cycle functions correctly. So, what they do is, if something's wrong with the cell, they will try and activate DNA damage proteins to come and repair the problem, or they'll make that cell die. It's a very rough world in this. So one of the most important genes we've already talked about actually is TP53. It's a gene that codes for the P53 protein, and it's located on chromosome 17. So I'm saying it's roughly around here in this diagram. I don't know about real life. And this is my take on what the protein could look like, just as an example. But what could happen is you get these mutations in the region. So what will happen is you form a defunct protein. So as you can see now, we've got these spiky edges, which might make it unable to perform its function that it's meant to have done. Alternatively, you can have loss in this region. So if there's a complete deletion, you won't get this section there. And so what you end up having is no protein formation. So another gene is ATM. It's located on chromosome 11. And again, my lovely take on what it might look like on PowerPoint. Again, if it's mutated, you'll get a really, you might get a different conformation of the shape, and so it's no longer able to do its job. Or you might not even get the protein to be made in the first place. So faulty proteins are not functional, and therefore the cell cycle cannot be enforced correctly. So what that means is abnormal cells can bypass those checkpoints, and then they can propagate. So what we tend to see is that these TP53 mutations are often seen in DEL17P patients, and ATM mutations are often seen in DEL11Q patients. But I want to say as well, as we've said, things are moving. So this is the biology behind it, but we are now coming up with different pathways, and so therefore new treatments are looking at how we can override these kind of checkpoints, I suppose. So one of the things that clinicians use is this cytogenetic technique called FISH. So it stands for fluorescence in situ hybridization. And basically it's a way of identifying if a particular sequence is present in your DNA. So I'm going to briefly go through it. So what you have is the sequence that you want. You then label it with fluorescent dye. You then manage to hybridize this into the DNA of interest, so a patient's DNA. And what should happen is you'll get that fluorescence popping up when you look under the microscope. So what ends up happening is if you're looking for that DEL17P deletion, if there is no fluorescence, that would suggest that nothing was able to hybridize, and therefore you wouldn't find any of these glowing points. So it's a tool that's really useful to help clinicians identify if and which chromosomes have changed. And then we can go on forward from there and identify the categories, or at least identify the treatments that patients can then go on to, should they need it. So again, this is just typical facts. So we've gone, so DEL13Q is typically associated with MCLL, and it's possibly the most prevalent form of CLL in populations. We've already talked about DEL11Q and DEL17P, and then there's also trisomy 12, which is it's probably the most common numerical abnormality in CLL, and this is a chromosomal abnormality, and it's something that I'm actually focusing on in my PhD. So that leads me nicely on to me, because it's all about me today. So I'm looking at epigenetics in CLL. So what we already know is that CLL is a B-cell cancer, and there are varied outcomes between patients. We have a number of patients here today, but the way your disease progresses will differ from someone else's. And this could be due to the genes that cause those problems, so differences in the genetics. But then it could also be due to external factors that impact the way gene expression takes place. So therefore, I'm looking at the epigenomics of CLL. This is how I looked when my supervisor told me this is what I'll be working on. But things have changed now. So I thought I'd quickly touch on gene expression. So what we have is DNA. We have the sequence that can then be read to form this intermediate molecule called RNA. And then this RNA is translated into functional proteins. And these are the things that make things work in your body. So epigenetics by the book is where you observe changes in organisms due to modifying how genes are expressed rather than altering the DNA sequence itself. So what we're saying is we're seeing how external factors alter the shape of DNA to allow or prevent gene expression. So here we have DNA that's fairly open, so gene expression can take place. And here we're having things that have bound onto the DNA. So the DNA sequence has not been mutated, but what's happened is you've changed the shape of the DNA, so now gene expression can't take place. And this is something that we were really interested in CLL. So I'm aware that some of you might not know still what I'm saying with epigenetics. So I thought I'd give you a little scenario. It's one of my favorite analogies, and I use it all the time whenever I can, and my mum and dad are sick of it now. So if we imagine that we're watching a film called Life, The Human Life, then the cells in our bodies would be actors and actresses. Wait, let's make this believable. So they'd be the actors and actresses following a script called DNA. There'll be points in this script that will have plot twists, and we can liken those to genes. Then our director, Epigenetics, can then change, cut, and put scenes together to make the final cut of the film. He's not changing the wording of the script, but he's changing what scenes would be played, even though the script might not want them to be played or whatever. So essentially, imagine if The Sound of Music was now directed by Steven Spielberg. It'd be a very different film. So one of the things that I'm looking at in my research is chromatin modelling. And basically what we have is this region which is closed, so no DNA is accessible. And here you have DNA that's accessible. So what ends up happening is these factors that can bind onto the DNA to allow gene expression can't bind in this region, but they can bind in these regions. So this one's coming up here. And what ends up happening is you'll get this, the DNA unwinding, and then you get that RNA molecule that I was just talking about, so that you can then make functional protein. So my work, if it comes up, because there are several subtypes of CLL, we want to improve our understandings of the interactions that the surrounding environment has on CLL DNA, CLL cells DNA, sorry. And then this way we can identify active genes in stimulated cells and then take them into the clinic as future targets for therapeutics. So we have methods already that can identify these open regions in chromatin. So essentially what I'll be doing is putting in factors into the blood, I suppose you could say. And then we want to see how that chromatin changes. And then maybe we can understand a bit more about the epigenetics of CLL. So I will look at these open regions using computational methods. And then we can validate these in the lab. And then we should hopefully see more prognostic markers that come up as a rise of epigenetic research. And because this technology is ever evolving, we're not just benefiting CLL, we're also benefiting other cancers and other diseases. So it's really good for many patients in the long run. So yeah, I want to thank my supervisor for tolerating me, and also the people at Bournemouth, and they're the ones who are funding me, so I have to make them impressed with my science. Thank you for listening. Ooh.